Couple of announcements. Brittany, are you recording this? Problem set 2? Yes, problem set 2 is due on Sunday night. Hard copies are available in the back and also on. If you left your Apple power adapter in the AMP lab, I have it. This is yours. This isn't mine. So if you're missing a phone adapter, this is my phone. This is your adapter. Okay. Today we are going to continue the visual system and figure out how we see, how we turn photons into vision. We're not going to get into perception too much, but a little bit. We probably won't have time to start with the auditory system. So this is aspiration. Let's start with some clickers. These are sort of pick-up clickers from the ones that I gave you as homework. Let's just start here. You are focusing your vision on a nearby object in bright light. What muscles are contracted? Both. You're in bright light, so your iris is contracting. Remember, it's a sphincter. Contraction narrows the opening. And you are focusing on a nearby object, which means you need a round lens, a bulgy lens, which means you have to relax the tension on the fibers that are holding the lens, because tension makes a flat lens. So you want to relax the tension to make a round lens, and that means contracting the ciliary muscle. Okay? All right? What nerves are firing action potentials to create this condition, to constrict these muscles? Oculomotor. I suppose there are action potentials going on in the other nerves, but the abducens nerve is what's used to, it's here, right? It's not out here. So we're not abducting our eyes. The cochlear nerve, I don't even know when. We use that in sort of a subtle way. So that was sort of a red herring distractor answer. The oculomotor nerve innervates both the iris and the ciliary muscle. Okay? You are focusing on a nearby object in dim light. Focusing on a nearby object in dim light. Let's assume that you've had a chance to adapt to the dimness of the light. What muscles are contracted? Ciliary muscles. Your iris is dilated, but your ciliary muscles are contracted to focus. It's difficult. It's not so easy to do. I mean, it's harder for you to focus in dim light on a nearby object than to focus on a distant object in dim light, because there is some coupling between the ciliary muscle and the iris. But you can focus on an object in dim light. It just takes a little bit more effort. What nerves are firing action potentials to create this condition? Pretty obvious. The oculomotor. Okay, so these are the sorts of things, I think, yeah, these are the sorts of things that you should be reviewing for the quiz that's coming up in a little over a week, and for the next exam. In addition to understanding the rectus muscles and the action of the abducens, nerve on the lateral rectus muscles, you know, the muscles we talked about, the anatomy of the eye that we talked about, the coloring book material about the eye. That's all coming up. But today what we want to do is talk about transduction, phototransduction. And that starts with the retina. The retina is the organ that we use to turn photons into a visual signal that our brains can interpret. And so there is oops, that's what it really looks like. Here's the cartoon. Here's the cartoon. Let's start here. The eye anatomy that you should know. We're focusing, we're zooming in on a little section of the posterior surface of the eye, including the retina. So that's what we're looking at here. The back of the eye. In fact, we're zooming in on a part of the eye that is near the fovea, where we have rods and cones. So at the back of the eye is the sclera, then the choroid, moving towards the front of the eye, sclera, choroid, pigment epithelium, and then retina proper. Okay? The layers of there are cellular layers in the retina. So let's talk about them in sort of the through path, the direct path of phototransduction. Photoreceptors are the next layer, and they're divided into two types, rods and cones. Rods are for black and white vision, high acuity vision, dim light vision, not color vision. Cones are for color vision, and our cones are mostly in the fovea. Rods and cones, photoreceptor cells, and we'll talk about phototransduction in a minute. The next cell type in the pathway is called the bipolar cell. There are synapses between photoreceptor cells and bipolar cells in this layer here, called the outer plexiform layer. That's not a name we need to know. So this is supposed to be cartooning synapses between a cone and its bipolar cell. Are rods and cones long or short receptor cell types? They're short. There's no axon. They don't fire action potentials. But they do form a synapse with all the goodies of the synapse, you know, calcium channels, synaptotagmin, vesicle, neurotransmitter release, receptors on the postsynaptic cell, in this case the bipolar cell. The next cell in the through pathway for light, photoreceptor cell, bipolar cell, ganglion cell. Ganglion cells are on the inner aspect of the retina, and they have axons. And these axons will form the optic nerve to carry the information from the retina to the thalamus in the brain. So that's the through pathway. Yeah?
So the bipolar cells are synapsing to the ganglion cells?
The bipolar cells form synapses on the ganglion cells, indeed. There is robust lateral inhibition in the retina. And there are cell types that do this robust lateral inhibition. The horizontal cells and the amacrine cells do lateral inhibition at the first synapse and at the second, and respectively at the second synapse. Horizontal cells do lateral inhibition at the first synapse, and amacrine cells do lateral inhibition in the second cells. So this is a way of sharpening receptive field, just like in the somatosensory system, but even more so. So here's the way it really looks. Let's get reoriented. So this is the outside of the eye, and the lens is that way now. Sclera, choroid, pigment epithelium, rods and cones, the cell bodies of rods and cones, called the outer nuclear layer. So this is what this really looks like. So the rods and cones, the outer segment of the rods and cones, and the cell bodies of the rods and cones. The synapses, the cell bodies of bipolar cells, the next synapse, the cell bodies of ganglion cells, and we're not seeing their axons. So what do you need to know? Sclera, choroid, pigment epithelium, rods and cones, the names of the photoreceptor cells, bipolar cell, ganglion cell. I'm not going to ask you to memorize inner and outer plexiform, although it's a fun word to say. Like I said, the fovea is where most of our cones are. So this is data to show you that that's true. So this is plotting the number of photoreceptor cells per millimeter, the density of each photoreceptor type, as a function of the angle from the fovea, where our best vision is. And at the fovea, in blue, you can see the number of cones. Very, very highly localized to the fovea. On either side of the fovea are the rods. And that's where our peripheral vision is good. Our dim light vision, it's important vision, but it's not sharp vision, and it's not color vision. Not very good color vision. This section going on an angle from the fovea happens to catch the blind spot where there are no rods and cones. The blind spot is where the axons of the ganglion cells go out through the back of the eye. Remember that the axons of these cells are here in the front of the retina, but the optic nerve goes out the back of the eyeball. There has to be a place for them to go. They collect. All those axons in yellow here collect at the blind spot and form the optic nerve that goes out the back of the retina. So each of our eyes, as I'm staring at Tynan's nose again, as I'm staring at Tynan's face, there's a blind spot. Each of our eyes is blind in a certain place, not too far from our fovea. But we don't see those blind spots. Our brain fills it in. So here is a cartoon of rods and cones. The outer segment is where phototransduction takes place. But it's a cell, so it's got a nucleus, and it's got a cell body, and it's got all those goodies that nuke cells do, and it's got a synapse, a very specialized synapse, onto a bipolar cell. Cones have the same sort of thing, an outer segment, cell body, and the specialized synapse onto their bipolar cells. In the outer segments of both rods and cones, there is a specialized membrane that contains the molecules that absorb light. In this cartoon, it's called pigment molecules. I'm going to call them opsins. In the rods, the opsins are in these discs that are inside the cone. Inside the rod. These discs of membrane. This is a membrane. This is a stack of membrane-lined discs. In the cones, it's not intracellular, it's part of the surface membrane. But the effect is the same. a large membrane area containing the molecules, the opsins that absorb light. Here is a cartoon, a structural cartoon of the membrane, so you recognize these guys. Here is an opsin. It's showing it as a stick model, but you're getting used to this. You can see that these are helices. If you count them, you'll find 7. So this is a GPCR. Opsins are G protein coupled receptors. Now you have come to think of GPCRs as always having a ligand that activates them. Light is not a, photons are not a very good ligand for molecules. But in the opsin is another molecule called retinal. This purple thing is retinal. It's a attached ligand. And light is going to be absorbed by retinal, and that's what's going to activate the receptor, and that's what's going to activate the G protein. The G protein, in this case, has a name. It's not GS, it's not GI, it's not GQ, it's called transducin. So in the photosystem, the G protein that is coupled to opsins is transducin. Just to be clear, rhodopsin is the opsin in rods. But we're going to see other opsins in cones. But the principles here are the same. Light is bound, or not bound, light photoactivates retinal, which activates the opsin, which activates the transducin. The pigment epithelium, the job of the pigment epithelium is to continually feed the rods and cones with new retinal. You may have heard about vitamin A. Vitamin A is important for vision. Vitamin A is important for retina. So
options are like photons?
no, opsins are like GPCRs.
Oh, okay.
Opsins are GPCRs.
That-
Retinal is like the ligand that is activated by photons. Photons are light. Okay? You all speak in the same language? So, one question is, how can one type of receptor give us color vision? Well, it's not one type of receptor. It's really 4 types of receptors that are very, very similar. We have slightly different opsins in the color-sensitive cones and in the rods. So let's start with rods. Rods have retinal. Rods have rhodopsin. It's got a name. And rhodopsin happens to have a sensitivity to light in the blue-green region. Right? So this is plotting sensitivity as a function of wavelength of visible light. Out here is ultraviolet. Down here is infrared, which we don't see. We humans don't see, but other animals do. Ultraviolet, which we don't see, but other animals do. You know, gamma rays and X-rays and stuff are out here somewhere. We see in the visible spectrum from 400 to 700 nanometers of wavelength. Rods are best in the blue-green region. That's the kind of light they detect best. They are relatively insensitive to purple and relatively insensitive to red, orange, yellow. in our cones, most humans are heterotrichromatic. That is to say, we express 3 different opsins. Each opsin is expressed specifically in a type of cone. So we have blue cones that express, the cones are not blue, blue sensitive cones that are that express an opsin that is highly activated in the blue-purple region of the spectrum. Other cones express other opsins. So for example, green sensitive cones that are not green, they are just green, sensitive to green light, they express an opsin that is highly activated in the green-yellow region of light. And we have so-called red cones, red sensitive cones that express a red sensitive opsin that is actually a misnomer because they are sensitive to red, but they are most activated by yellow. So you can see that, deep red light is activating only the red cones. Deep purple light is activating only the blue cones. Other colors are activating a range of opsins, and that's how we differentiate color. People who are colorblind have a genetic abnormality where one or more of these opsins are not expressed. So for example, I'm pretty sure that people who are red-green colorblind do not express the red opsin, and so it's very difficult for them to differentiate between green and red. All of these, the photopigment, the thing that actually absorbs the light of various wavelengths, is still retinal, but what is different are subtle amino acid replacements in the opsin that change the sensitivity of the retinal to light and give it this spectral, give it each opsin with its retinal a spectral range. So this is the receptor environment is changing the absorbing properties of the retinal that's embedded in it. There are some humans that are heterotetrachromatic, that have a 4th opsin. Who knows, maybe those are highly evolved humans and we are all eventually evolving towards heterotetrachromatic vision. But right now, most of us are heterotrichromatic, three cones and 1 rod opsin for rods. Questions about how we see color. This is how we see color. So now let's talk about how we see light in general. So we're talking about a cone, just because, but the same thing is going to be true in a rod. The opsin, with its embedded retinal, is highly expressed in these folds of membrane, whether the folds of membrane are surface membrane in a cone or in the discs of a rod. High density of opsin with its bound retinal. High density of the transducin that is linked to the opsin. Remember, what gives us color vision is a difference in the opsin, not a difference in the retinal or the G protein. So let's start in the dark. We're in the dark. In the dark, this system is not working, but there is activity. There is a lot of activity. In the dark, an enzyme, not adenylyl cyclase, it's called guanylyl cyclase, because it makes cyclic GMP. Okay, it's just like adenylyl cyclase except that it's guanylyl. Turns GTP, which you've heard of, into cyclic GMP, which you may not have heard of yet, but now you know. Guanylyl cyclase is also highly expressed in these folds of memory, in the outer segment of rods and cones. So in the dark, we are making, our rods and cones are working hard to make GTP, to make cyclic GMP. Cyclic GMP activates a cyclic nucleotide activated ion channel. So in the dark, we have a lot of sodium influx into photoreceptor cells. So in the dark, do photoreceptor cells have a resting membrane potential that is hyperpolarized or depolarized relative to what you think of as a normal cell, even a normal neuron? Depolarized, because we have a lot of sodium influx through these channels, through the cyclic nucleotide gated ion channels. So the rods and cones have a relatively depolarized resting membrane potential in the dark. And this is called the dark current by biophysicists, for obvious reasons. Okay, now, let there be light. We're now in a light situation. Light hits the retinal, causes a cis to trans isomerization of the retinal. So there's the first step. Light, cis trans isomerization of retinal, conformational change of the opsin, conformational change of the G protein, transducin. You know what happens next. Cyclic, what happens when we activate a G protein? GDP dissociates, GTP associates. We now have an active G protein. The alpha, so this doesn't show it, but it's the alpha subunit, the activated alpha subunit of transducin activates an enzyme called phosphodiesterase. This is the effector of this pathway. So this is transducin as the heterotrimer, but remember it's the alpha, it's the GTP activated alpha subunit that activates phosphodiesterase. Phosphodiesterase, its name tells you what it does. It cuts cyclic GMP into GMP. It breaks down the cyclic GMP that you made during the dark. Well, this is weird, but this is the way it is. Breaking down cyclic GMP lowers the concentration of GMP, which causes the cyclic nucleotide gated ion channel to not have its activator, so it closes. So light causes the closing of a sodium-calcium channel. What does that do to the membrane potential? Hyperpolarizes it. Light causes a hyperpolarization of the rod or cone. What is different about these three sweeps, these three traces? So here we're plotting membrane potential, voltage, as a function of time after a pulse of light is given to a rod or a cone. Here's the relatively depolarized resting membrane potential, and here's the hyperpolarization in response to light. What do you suppose is different about these three? The brightness of the light. This is a graded response. A brighter light produces a more hyperpolarized membrane potential in the rod or cone. So, just like in other cells, a depolarized membrane potential leads to neurotransmitter release, and a hyperpolarized membrane potential leads to less neurotransmitter release. And that's what's gonna happen at this synapse. In the dark, we have a lot of neurotransmitter release. And that's why this synapse is specialized. It's specialized for constant darkness-induced neurotransmitter release. But in the light, the hyperpolarization causes a decrease in neurotransmitter release. And so when we see light, the first thing that happens at the first synapse is a decrease in neurotransmitter release. It seems upside down, but that's the way it is. So now we have to talk about, so we're done with the retina. No, sorry. We're done with rods and cones. We're still in the retina. Light, and all the steps that I just told you about, cause a hyperpolarization that cause a decrease in neurotransmitter release. Okay? Now we have to turn that simple response into vision. So we got our work cut out for us, because right now things are way too simple. In the retina, we have two types of ganglion cells. Okay? So now I'm jumping from the rod to the ganglion cell. And remember, it's the ganglion cell that carries information from the retina to the brain. Okay? So I'm jumping around a little bit, but bear with me. There are two types of ganglion cells. There is the so-called on-center, off-surround ganglion cell. What does that mean? It means that when light is shined on the center of that ganglion cell's receptive field, we get an increase in the frequency of action potentials. That's what this is supposed to illustrate. This is action potentials, and this is an increase in the frequency of action potentials in response to light on the center. Light on the surround, on the periphery of this ganglion cell's receptive field, inhibits action potentials, decreases the frequency. So that's why this is called an on-center, off-surround ganglion cell. Light in the center, more action potentials, light in the surround, less action potentials. We're going to focus on understanding the on response. So we're not going to really focus on the center response. Let's focus on the center responses and not worry about the surround responses. But it'll all fit together, at least I hope it will, when we get there. So one type of ganglion cell is the on-center. Well, predictably, another type of ganglion cell is the off-center, on-surround. This is a different type of ganglion cell where light on the center of that ganglion cell's receptive field decreases action potential frequency, and light on the surround of that ganglion cell increases action potential frequency. But let's focus on the center response. So this would be called, for us today, an off-center ganglion cell. So two types of ganglion cells. Ganglion cells whose action potential frequency is increased by light on the center, and ganglion cells whose action potential frequency is decreased by light on the center. By center, I mean center of the receptive field of that ganglion cell. Okay, this is weird, so we need to understand it. And we're going to understand it with this cartoon. So in your handout, you have one snapshot, but my recommendation is to use that snapshot as a reminder to go to Moodle and look at it in steps. So that snapshot is going to be is the final step of a story that is confusing, but let's go through it. So here is, let's walk through this. Here's the photoreceptor cell. This is a cone. It has a synapse. It synapses with bipolar cells. It synapses with two different types of bipolar cells. So now I'm not skipping the bipolar cell. I'm reintroducing, reminding you of the bipolar cell. And these bipolar cells interact with two different types of ganglion cells. And you can see where this is going to end up. One is going to be an on-center and the other is going to be an off-center. You've got to figure out how that happens, but that's where we're going. Okay? Light. So let's just walk through this. Light. Let's measure the membrane potential. What does the membrane potential do when we turn on light? It hyperpolarizes. Why? Because the cyclic nucleotide gated channels are closing, because cyclic GMP is being hydrolyzed, because phosphodiesterase was activated, going backwards in time. Hyperpolarization of the photoreceptor cell. What does that do to the release of neurotransmitter at this synapse? Decreases it. Less neurotransmitter release at this synapse. So in the dark, there was a lot of neurotransmitter release. In the light, there is less. What is the neurotransmitter at this synapse? This is key. The neurotransmitter at this synapse is glutamate. You need to know that. So light causes less glutamate release. Okay, now here is the key to understand right now. On the two different types of bipolar cell are two different types of glutamate receptors. There are AMPA receptors on one type and metabotropic, not muscarinic, metabotropic glutamate receptors on the other type. AMPA receptors are stimulatory receptors. They activate cells. And metabotropic glutamate receptors don't. They inhibit cells. And that's what's supposed to be. So on this type of bipolar cell, there are AMPA glutamate receptors that activate this neuron, depolarizing it. And on this neuron, this bipolar cell, it expresses metabotropic receptors that inhibit, that hyperpolarize this and inhibit it, and cause it to not fire, well, to become hyperpolarized. So let's go back. In the dark, there is a lot of glutamate, which is causing a depolarization of this cell, and not a depolarization of this cell. In the light, less glutamate, less depolarization of this cell, and more depolarization of this cell. So how did that work? Less neurotransmitter, less activation, that's easy, less inhibition, which equals activation in this cell, the double negative. Inhibition of inhibition. You don't need to know 6, you don't need to know kainate. All you need to know are AMPA receptors that are ionotropic and metabotropic, which are GPCRs that are inhibitory. That's what you need to know on the two different types of bipolar cells that are synaptically coupled to this cone. So let's record from this cell. What's our prediction? When light hits this photoreceptor cell, what's going to happen to the membrane potential of this cell? Is it going to depolarize or hyperpolarize? Depolarize. It doesn't have sodium channels, it doesn't fire action potentials, but it does depolarize. Okay, let's record from the other cell. What's going to happen to the membrane potential of this cell that's expressing AMPA receptors when light hits this cell and causes less glutamate release? What's going to happen?
Like,
It's going to hyperpolarize. So this is the key to understanding on and off center. Right there. And since your slide, your handout, has them both sort of embedded and it's not color, it's hard to tell. But walking through this series of images on the Moodle will help. Okay, so we've gotten to these bipolar cells. The type that expressed the inhibitory receptor depolarized in response to light, and you can imagine that that light causes an increase in neurotransmitter release from that bipolar cell on the next synapse. And the other cell that expressed the excitatory receptor was hyperpolarized by light, hitting the photoreceptor cell. And so that hyperpolarization causes a decrease in neurotransmitter release at the next synapse. So now let's record from this ganglion cell. Oh, I need to tell you. The neurotransmitter at this synapse is glutamate. Again. These cells express ionotropic glutamate receptors. So what's going to happen to this cell? It's going to depolarize, and this cell has an axon. So what's going to happen to its action potential frequency? Increase. Light on the photoreceptor cell causes an increase in this ganglion cell's action potential frequency. Now you can see where this is going to go. The other type of ganglion cell is going to have the light is going to inhibit the frequency of action potentials. It's going to have action potentials when this cone is in the darkness. In the darkness, lots of neurotransmitter release, lots of depolarization, lots of neurotransmitter release, lots of action potentials. In the darkness. In this cell, in the lightness, less inhibition, depolarization, lots of neurotransmitter release, lots of action potentials. So now you understand how there are two types of ganglion cells. The ganglion cells are not different. It's the bipolar cells that are different. What's different about the ganglion cells is which type of bipolar cell they're connected to. Complicated. But you tell yourself the story enough times and it becomes easy. Are there any questions? Before we're going to leave the retina, because now we have action potentials, either an increase or a decrease in action potential frequency. We have action potentials leaving the retina and going to the brain, and that's the signal that we see.
Could you go over the double inhibition part again?
Sure. In the dark, this cell has a depolarized resting membrane potential because of the open cyclic nucleotide gated ion channel. So in the dark, this cell is releasing a lot of neurotransmitter. This cell is expressing an inhibitory receptor. So a lot of neurotransmitter in the dark is inhibiting this cell, causing not very much neurotransmitter release here, not very much action potentials here. In the light, hyperpolarization, less neurotransmitter release, less activation of the inhibitory receptor, less inhibition allows depolarization, more neurotransmitter release, more activation of this receptor of this cell in the light. So this cell becomes an on cell, and this cell becomes an off cell. Yeah.
The graph for the ganglion cells, is that showing that it's in the dark?
Well, it's showing what's happening in, you're talking about here?
Yeah,
The
so
graph.
the one before when I had the yellow. That was showing in the dark
In
because
the-
those are the action potential.
In the light, this is this cell, right? In the light, action potentials are not on, and in the dark, action potentials are on. And that makes this an off-center cell. So it gets confusing which is which until so you have to go backwards in time to know that the first one is the on-center cell and the second one is the off-center cell. That's why this graphic gets confusing when you have only one picture of it. So walking through this stepwise on Moodle is your key to remembering. Other questions? Okay, we have action potentials, either more or less. Remember that fewer action potentials is a signal. That's a signal too. Just like hyperpolarization is a signal. So hyperpolarization is a signal, fewer action potentials is a signal that our brain interprets. So now we're going to go into the brain. What we have done is created action potentials, either more or less, in the optic nerve. Now, I'm not going to say it. I'm trying to avoid saying the thing that is wrong. But I know that by saying the thing that is wrong, sometimes that puts the wrong information into your brain. So what is right is that your left visual field, so right now I'm looking at Brittany, my left visual field is going to my right brain. And my right visual field is going to my left brain. How does that happen? It happens at the optic chiasm. So now let's just look at this. Here are two eyeballs. This is looking at eyeballs from above. Here's the nose. Here's the longitudinal fissure. Right here is the blind spot, where axons go out the back of the eyeball. On either side, but more importantly, it's also, in this cartoon, it's also where the fovea is. The blind spot is not the same as the fovea. The blind spot is the opposite of the fovea, but let's think about this as the fovea. Right visual field, left visual field, fovea, right? Right, left visual field, all my peripheral vision, but I'm staring at the oak tree out the window. So let's think about the right visual field. When I stare at this oak tree, I have visual information coming from my right visual field to both of my eyes. The right visual field of my left eye is mapped laterally because the lens, crosses it. The right visual field of my right eye is mapped medially on the retina. So different parts of the retina are mapped, are activated by the right visual field. The right visual field activates the left or lateral part of the left eye and activates the right or medial part of the right eye. And the left visual field activates the medial or right part of the left eye and the lateral or right part of the right eye. Okay? So getting this image into your brain is going to be important for understanding this. Axons from the lateral portion of the eye do not cross. axons from the lateral retina do not cross. So axons from the lateral left eye end up in the left lateral geniculate, thalamus. And axons from the right part of the right eye end up in the right lateral geniculate. On the other hand, axons from the nasal or medial part of the retina cross. So axons from the medial part of the right eye go to the left lateral geniculate. And axons from the medial or right part of the left eye go to the right lateral geniculate. So the thing that is wrong, that you may have heard, is that your right eye is mapped to your left brain, and that's wrong. So forget that. That's what you need to forget. What you need to remember is that the left visual field of both eyes is mapped to the right brain, and the right visual field of both eyes is mapped to the left brain, and this is how it's done. I do want you to know the name of this nucleus in the thalamus, the lateral geniculate nucleus. That's the part of the thalamus that is dedicated to processing visual information. And then there's a bunch of synapses in the thalamus, in the lateral geniculate nucleus of the thalamus, and then information is going to go to the visual cortex. And you remember where the visual cortex is. It's in the posterior occipital lobe. Remember, the left occipital lobe is processing information about the right visual field. And the right occipital lobe is processing information about the left visual field. And luckily, we have a corpus callosum that is telling our brain, giving it, the brain, the information is getting to both sides, but not yet. So let's take a short break, because this has been a lot of material. And we'll pick up here. So I need to go backwards a little bit. I'm going to go back. So I can start here. So this is the ganglion cell layer. So the vitreous humor is down here, and the lens is in the subbasement somewhere, right? So the light is coming up, and the light has to go through these layers of cells before it activates its receptor. These cells are relatively transparent. This is an argument against intelligent design, because it's not designed very well. I mean, this is what we evolved. And the reason we evolved it is that we need one of the arguments for how this evolved is that we need a non-transparent layer of nutrient. The pigment epithelium needs to not be where the light is, because it's not transparent. So we put the nutritive layer away from the light, then the light detecting cells that need to be in contact with the nutrient layer, and then all these processing cells in order to process information in the retina. You could come up with a better system, but this is how we evolved. So the fact is, light has to go through layers of cells to activate the ganglion cell, and then the axons, which have to go out the back. So they loop-de-loop out the back, forming that blind spot. This is an image that I need to tell you just because this is a drawing from Santiago Ramon y Cajal. So Ramon y Cajal is credited with being the father of cellular neuroscience. And remarkable insight. He was able to put this together. So here is an image of an arrow being projected on both both the left and the right visual field. And he was able to put together that the right visual field is mapped to the left occipital lobe, and the right visual field is mapped to the left visual field is mapped to the right visual cortex. So, amazing insight. I don't talk a lot about experiments that lead to our understanding in this course, because it's too much to talk both about the information and where the information comes from. But this is a critical one. Santiago Ramon and Call figured this out. Just want to mention the lateral geniculate. I've mentioned it before. The lateral geniculate nucleus of the thalamus is organized into layers. You don't need to know the details of the layers. Three of the layers receive information from the ipsilateral eye, and 3 of the layers receive information from the contralateral eye. Remember, the left lateral geniculate is processing the right visual field from both eyes. The left lateral geniculate nucleus is receiving information from both eyes, but it's segregated into different layers of the left lateral geniculate nucleus. And there are different cell types in the lateral geniculate nucleus that allow us to process information differently. So I don't want you to memorize this information, but I do want you to just be amazed that in the primitive centers of your brain, you are already starting to figure out, okay, is the information that I'm getting moving, or do I need to get information from it regarding its color and its shape, right? So even in the thalamus, we're starting to process information regarding movement and sort of broad information versus acuity, sharp and color information. But let's go to the visual cortex. So from the thalamus to the visual cortex. But before I do that, I want to mention one more thing. One is that information from the eye goes to other areas of the brain besides the visual cortex. It goes to the hypothalamus to help control your diurnal rhythm. Most of you are on generally a day-night cycle. You sleep at night and you are awake during the day. Most of you, most of the time. Some of you are on a completely nocturnal schedule. And being here this morning is difficult, but that's not typical. So, regulation of your circadian rhythm, control of the dilation of your pupil, focus, is not done in the visual cortex, is done in something called the pretectum. I don't want you to memorize that name. The superior colliculus, which we saw on the sheep brain, and we will maybe see on the human brain, is important for orienting the eyeballs, controlling the rectus muscles and the oblique muscles to orient the eyeball so that the fovea of both eyes is focused on the same object. So there are other parts of the brain that are essential for vision besides the visual cortex. And here's the interesting thing. Some of this information to this part of the brain doesn't even require photoreceptor cells, because there is a class, and I will never test you on this, there is a class of ganglion cells. There is a class of ganglion cells that are intrinsically photosensitive. They're called intrinsically photosensitive ganglion cells. We do not use them for vision, but we use them for processing of light information to do things like this. Circadian rhythm, reflex controls of pupil and focus, movement of head and eye. Okay, so now let's let's go to the visual, let's get back on track with vision. What do we see? We see in the first part of our cortex that sees is our visual cortex. Posterior occipital lobe. Posterior occipital lobe. Above and below the calcarine sulcus. This gyrus has a name, this gyrus has a name. I don't care that you memorize those names, but I do want you to know the name of the sulcus on both sides of that divide the visual field, that divide the visual cortex vertically. So this is a midline sagittal section of the brain, anterior, posterior, superior, inferior. So this is a right hemisphere. So what visual field is this hemisphere processing?
Right.
The left visual field, which is being processed in the gyrus, in the gyri, above and below the calcarine sulcus. Okay, here's the cartoon. This is a midline sagittal section, right hemisphere, left visual field, calcarine sulcus, the gyrus above and below the calcarine sulcus. Okay? Now what is the point I'm trying to make here? Our visual field is a two-dimensional surface, and our visual field is mapped onto the neurons of our primary visual cortex above and below the calcarine sulcus. So just like in the somatosensory cortex, your body is mapped onto the post-central gyrus. In the visual field, the visual field is mapped onto the gyri above and below the calcarine sulcus. The center of the visual field is mapped posterior, and the periphery of the visual field is mapped anterior. The part above the fovea, remember this is all left visual field, because we're in the right brain, right hemisphere. The part above the visual field, above the fovea, 1 and 3, are mapped to the gyrus below the calcarine sulcus. And the part of the fovea, the part of the visual field below the fovea are mapped to the gyrus above the calcarine sulcus. So that's a mapping. I do want you to understand the two-dimensional mapping of the visual field. Center, posterior, superior, gyrus below. That's the map. That's the details of. That's the amount of detail of the visual field mapping that I want you to know. So just like knowing that the somatosensory cortex, the toes are mapped medially on the somatosensory cortex, I want you to know that the fovea is mapped laterally or posteriorly on the visual cortex and things around that. Okay, is that clear? From the visual field, no, say that again. From the primary visual cortex, information is processed by the rest of the brain. So the primary visual cortex is essential for turning images into vision, but it's not the only. Our primary visual cortex is good at picking up. What our primary visual cortex is best at is picking up edges. Edges of objects. But obviously that's not all we see. We make an image and we see because we can put together images. But it's not all in the visual cortex. The visual cortex starts by making information related to edges of objects, and then the rest of our brain is important for filling it in. So there is the ventral information flow from our visual cortex into our temporal lobe that provides information about what is that thing I'm looking at. Gives it a name. And the dorsal stream from the occipital lobe into the parietal lobe provides information about where is it and is it moving? So, for example, you know, somebody is coming towards me, and I see that object moving towards me. And I'm processing information. Oh, that's a friend. That person's moving towards me. I'm going to get ready for a hug or something, or a positive interaction. Oh, no, that's a bear moving towards me. I better get the hell out of here, right? So different kinds of information are processed in different, and then the corpus callosum is putting things together on the two hemispheres. But I don't want you to be left with the idea that vision stops here. Vision is still really primitive here. It's when we get vision, what we see is a whole brain thing. It's a perception is not a part of the retina. It's not a part of the thalamus. It's not a function of the primary visual cortex. It's a function of all of those things plus other parts of the brain. So remember, you know, what is it is going to the temporal lobe, and then giving it a name is probably, you know, part of our frontal lobe. But I do want you to know the two visual streams from the occipital lobe. The ventral stream to the temporal lobe, what is it? The dorsal stream to the parietal lobe, where is it? Okay, so so vision is a, can you lower the screens there? Vision is a, we construct vision. We have a blind spot, we don't see the blind spot, we're filling it in. Our brain fills it in. There are some good animations, but let's just play around with this. I want to use some optical illusions to demonstrate how vision is a construct. You don't have a photograph of me in your brain. You have a construct of me in your brain. So here is an optical illusion. You stare at this spot. It looks white. But in your peripheral vision, these other white spots are looking not white, right? So I don't know about you, but if I stare at the center spots, the spots in the periphery are gray and sometimes even black. But then when I look at them, oh no, they're not black, they're white. So depending on where you put your fovea, there's an illusion here. This is a well-known illusion, and the so-and-so grid illusion, where what is white looks black in your peripheral vision. You see what I'm talking about? Here's another optical illusion. So these two tiles look like they are different color. Would it surprise you to know that this gray right here and this gray right here are exactly the same gray? You don't believe it. You say that's not possible. This is a white tile, and this is a gray tile, so there is no way that this gray and this gray are the same. But look what happens. I promise you, all I'm doing is putting up a screen. I'm just putting up a black box on the interface. Remember, you're best at seeing edges, and then your brain fills it in. So now you start to say, okay, I've blocked out the edge. You don't know that this tile is a different color from this tile anymore. Although you might be remembering it, And you might be saying, well, this tile is still looking a little lighter than this tile, but that's because the sky is up and the ground is down. All I did is flip it upside down, and now they look very much, for me anyway, this color right here and this color right here, it's the same color. It's only because your brain tells you that they're not the same color, that they look not the same color. Vision is a construct of what you expect to see. Would it surprise you to know that square B is exactly the same color as square A? I can't show it to you, but if you watch either of these videos, you will see this optical illusion happening in front of your very eyes, that this tile and this tile are the same color. You don't believe it because you know, okay, this tile is in this cylinder shadow, but it's a white tile, so this color is the same as this color, but it's not. This color is the same as this color. You don't believe it, but watch the video. Vision is a mental construct of our experience, what we expect to see. So this is a cartoon. No, it's a drawing. It's artwork. What you see it depends on your experience, your expectation, the circuits in your brain. So now I'm going to tell you that when I reveal the image on the artwork, children under the age of 8 see a bunch of dolphins. Their first response is, oh, dolphins, fishies. They're not fish, but dolphins. Adults typically do not see dolphins until they work at it. They have to work at seeing the dolphins. So I'm going to show it to you. You can decide what you see first. Adults typically they don't see the dolphins. They see the human shapes. But they're dolphins, right? You can oh, there's the dolphin. There's a dolphin. Yeah, I see the dolphins. But what you see first is a function of what you know, what you might expect to see, what you want to see. Okay? So so we're done with vision. I just want to point out that in physiology, vision is simple, but in perception, vision is complicated. There's a great video. So I'm going to play the video to just get your appetites whetted so we're going to, I'll just come on Tuesday. Come tell your friends to come on Tuesday because we're going to start the auditory system, and we're going to start with a really good video to get the auditory system in going. So I don't want to mess up your knowledge of the visual system. We have a couple of minutes. Are there any questions? Okay, well, use your couple of minutes productively. I'll see some of you in lab this evening. And the rest of you, I'll see you on Tuesday.